Now, DNA protein interaction can be studied by, you all know, this is suppose DNA, this is a protein, this is a steroid hormone receptor. Steroid hormone receptor. Yes, you must have read steroid hormone receptor in pharma, in physio. Steroid hormone receptor act on the DNA, and we know that steroid hormone receptor bind to the DNA and inhibit or activate the transcription. Yes, that you already know that in lacoprone. I discussed lacoprone. Yes, at that time I told you that lacoprone, this cap cyclic AMP bind to capside, repair tetramer bind to operator site. And I told you repair tetramer bind to operator site and inhibit the transcription. Cap bind to capside and increase the transcription. So any protein, yes, cap cyclic AMP protein will bind and will activate transcription. Repair tetramer will inhibit the transcription. So in this way, we found that few protein activate the transcription, few proteins inhibit the transcription. So you will say in this way, sir, any protein would come and will activate the transcription, any protein would come will inhibit the transcription. No. Yes, only those protein can bind to the DNA which have a motive. Suppose a protein came to DNA and want to bind to DNA. DNA will ask तेरे पास क्या है क्या? Yes, what do you have? Yes, तेरे पास क्या है क्या? Protein कहेगी मेरे पास माँ है माँ. Ma means motive. Yes, Ma means motive. Only those protein can bind to DNA which have motive. Motive may be zinc finger motive, helix turn helix motive, helix turn helix motive. Yes, zinc finger motive, helix turn helix motive. Zinc finger motive, helix turn helix motive. Okay, so we found that zinc finger motif or helix turn helix motif, yes, steroid hormone have zinc finger motif. What is motif? Motif is that part of protein which directly bind with DNA. So we found that steroid hormone receptor have finger-like projection, and in these finger-like projection we found that zinc is there, and these are called zinc finger motif, finger-like projection, and these finger-like projection make direct contact with DNA. So DNA protein interaction is mediated by motif, and we found that if you want to study DNA protein interaction, this can be done by DNA footprinting. What is DNA footprinting? No question as such in detail, but you can simply remember in DNA footprinting, we found that whenever DNA and protein interact, and when any enzyme just like restriction endonuclease want to cut this. Suppose this is normal DNA. Restriction endonuclease can cut at this side, can cut at this side, that can at this side. But if motif is already attached, protein is already attached, then it would not be cut at these sides. And if you will do electrophoresis, if DNA without protein, the fragments would be different. If you do electrophoresis with DNA, yes, having motif, having protein, the electrophoretic bands would be different. So you can determine the DNA protein interaction by DNA footprinting. Yes, I deliberately took this zinc finger motif. This is looking just like a duck, and this is looking just like the foot of duck. Yes, so now you can easily remember DNA protein interaction is studied by DNA footprinting. Understood? So this is about now PCR, polymerase chain reaction. I want to amplify DNA in test tube. I want to amplify DNA in test tube. First question is asked, what you will take in test tube. First thing, I will take the DNA of interest. Suppose this is DNA which is to be amplified. First thing I will do, yes, first step is denaturation of DNA. Heat. You will heat the DNA. When you will heat the DNA will be separated. DNA will be separated. Second thing, you will do re-annealing. What does it mean? Means first thing, you added DNA of interest which is to be amplified. Second thing, you will add two types of primer. How many primer? Two types of primer, no two primers, two types. One primer for this strand, one primer for this strand, one primer for this strand. Two types of primer, and these primers are not RNA, these are DNA. These are DNA. We add deoxynucleotide because we are synthesizing DNA. We will add deoxynucleotide. Yes, yes, these are not, yes, deoxynucleotide, these are not dideoxynucleotide. Okay, sorry, helix turn helix motif is for lacoperon. Helix turn helix motif, helix turn helix motif, lacoperon means lac proteins. All lac proteins means lac I, lac pressure protein, cap catabolic activated protein, both are having helix turn helix motif. This is test tube. I want to amplify DNA in test tube. If you remember the steps in DNA synthesis, you remember, yes, I don't turn pages, you will tell me, and you know that first of all what we do, double stranded DNA we separate, we put helicase move, two strands are separated, it produce supercoiling. To reduce supercoiling, to remove supercoiling, we have topoisomerase 1 and 2. We cut with the help of topoisomerase 1 and 2. But this problem can be solved by PCR. In PCR, only by heat. If you heat, two strands of DNA will be separated, no supercoiling. So helicase, the function of helicase and topoisomerase can be taken care by only and only heat. Two strands are separated. When two strands are separated, we require primer to synthesize, but we can't keep all the enzymes in test tube. We are amplifying DNA in test tube. Actually, when we do DNA synthesis in the cell, we have all the component in the cell, all the enzyme in the cell. These enzymes work in a sequence, but yes, we found that, in test tube we can't determine the sequence. That's why we want to take only one enzyme, and that would be DNA polymerase. All other enzymes will be compensated by any other mechanism, just like I compensated helicase and topoisomerase. That function is taken care by heat. Okay, now we want primer because third enzyme was primase. We can't take primase in the test tube because, you know, in test tube we can't determine the sequence of enzyme. That's why we took primers, already synthesized primer, two types of primer. Yes, and these primers are DNA, not RNA primer. Now we third thing, we will add deoxynucleotide to synthesize DNA. We also require deoxynucleotide, and we take Taq polymerase, thermophilus aquaticus polymerase. Yes, this is warm water bacteria, it can tolerate heat. Yes, fifth thing, we take magnesium act as buffer, it maintain buffer system. So these five are component of PCR we take in test tube. Yes, now we can start DNA synthesis. We took the target DNA, we heated, denaturation, two strands are separated, that re-annealing, when we decrease the temperature, then these two strands bind with primers. Then third is extension. extension. What is extension? Means Taq polymerase will come and will extend it. Taq polymerase will come, will extend this. So these three steps are there in DNA, in PCR, and DNA is synthesized. So one step is denaturation, we heat it, two steps are separated, the function of helicase topoisomerase is taken care. After that, primase, we don't want to put primase who will determine the sequence of enzyme in test tube. That's why we took two types of primer, preformed primer, one for each strand, and when we decrease temperature, primer attached to them. Then extension, then when the Taq polymerase come and extend these primers and DNA synthesis occur. In this way, 1 to 2, 2 to 6, 2 to 4, 4 to 8, 8 to 16 exponential growth occur, and we found that after 20 cycles, 2 to the power 20, around 10 to the power 6, millions of copies. After 30 cycles, 2 to the power 30, 10 to the power 9 billion copies. Means after 20 cycles, millions of copies. After 30 cycles, billions of copies of the target DNA is produced. Yes, so this is PCR. In a short time you will get multiple copy. But what are the limitation of PCR? One limitation is you can amplify short segment of DNA. First, not a long segment of DNA cannot be amplified. Second limitation is these are called, yes, we are making primer. If you want to make primer, you must know the nucleotide sequence. Yes, flanking sequence. This sequence is called, suppose this target DNA is to be amplified, you must know the nucleotide sequence near to the target DNA. This is called flanking sequence. This is called flanking sequence required to make primers. So if you don't know flanking sequence, you can't do PCR. If you want to do PCR, you must know flanking sequence, because these are required to make a primer. And a small fragment of DNA can be amplified. In a short time you can find multiple copy of DNA. As I told you, billions of copies of DNA after 30 cycle. But suppose patient came, you took sample, you put all these things in a test tube, and you started PCR is going on. After some time you came again and you thought that now I should have sufficient copy of the target DNA, but due to any reason, due to impurities, this PCR is stopped in the middle. PCR is stopped in the middle. PCR is stopped in the middle. Now what will happen? That's why we want to monitor the PCR in real time. Means we added one more thing. We added cyber green dye. SYBR, cyber green dye. This give fluorescence when attached to double stranded DNA. Cyber green dye don't give fluorescence, but it give fluorescence only when attached to double stranded DNA. Okay, I leave this discussion. Yes, what is flanking sequence? Suppose I ask you, amplify this DNA. This DNA. I give you this DNA to amplify. I can amplify only this DNA if I know the nucleotide sequence at this point end. If I don't know these sequence, I can't make primer for that. So the means we say that the target DNA which is to be amplified, the end portion, flanking portion, nucleotide sequence must be known to you. Otherwise you will not be able to make primer. Got the point? So flanking sequence, whenever a DNA target DNA is to be amplified, we think that okay, what is this DNA? Flanking sequence should be this. If we don't know flanking sequence, we can't do replication of DNA in PCR. So this is limitation. So PCR was going on due to any impurity. Suppose PCR is not going on, that's why we always monitor the PCR in real time. What we added on this PCR, if I add one more thing, cyber green dye. Cyber green dye give fluorescence when attached to double stranded DNA. Normally it don't give, but when attached to double stranded DNA, it give fluorescence. So what happened? Sixth thing, I attached this, I added this thing. Now that this PCR, normal PCR becomes real time PCR. What does it mean? Means whenever one DNA will be replicated, two copy, two cybergreen dye will bind, fluorescence will increase. 2 to 4 copies, 4 cybergreen dye will bind to double stranded DNA, will show fluorescence, fluorescence increase. 4 to 8, fluorescence increase. 8 to 16, fluorescence increase. 16 to 32, fluorescence increase. So if at any point of time I ask you how much DNA has been formed, you will look at the fluorescence, you will look at the fluorescence and you will tell yes, this amount of DNA has been synthesized till now by looking at the intensity of fluorescence. Means as PCR will flow, yes, forward, yes, move forward, the fluorescence intensity increases. Means you can monitor the PCR in real time. At any time, if I ask you the PCR is going on or not, you will say yes, I can tell you. How much DNA has been synthesized till now, you can tell me yes. And how much means quantity. That's why real-time PCR is also called quantitative PCR. This is called quantitative PCR. Understood flanking sequence, understood real-time PCR. PCR in real-time PCR, extra thing is cybergreen dye. As you add cybergreen dye, as this PCR will go 1 to 2 copy, 2 double stranded DNA bind to cybergreen, fluorescence will increase. 2 to 4, 4 to 8, fluorescence will increase. At any point of time, if I ask you how much DNA has been synthesized till now, you will look at the intensity of fluorescence and will tell me that this is the concentration of DNA synthesized till now. Means you are quantifying the result. That's why quantitative PCR, you are monitoring the PCR in real time. That's why it is a real time PCR. Now a frequently asked question of MLD, once asked in our exam also, they give nucleotide sequence of DNA, flanking sequence, okay, and they ask the nucleotide sequence of primer. Just like they gave the DNA sequence to be amplified PCR is shown below. The dashed line indicates the internal sequence where the potential mutation is located. Means mutation is located over here. Means we want to amplify this portion. And primer, they ask, yes, two types of primer. So mnemonic for this. First of all, mnemonic. Whenever they ask about the primers, write both strands in 5 to 3 directions. Write both the strand in which direction? 5 to 3 direction. Yes, 5 to 3 direction. Both strand in 5 to 3 direction. Okay. They have given one strand in 5 to 3 direction, another strand 3 to 5. So I will write the lower strand in 5 to 3 direction. And I would say that these initial two nucleotide sequence are sequence of primer. This may be a mnemonic, this may be a concept, whatever you consider it. But you will remember whenever a nucleotide sequence is given, double stranded DNA is given, you will write both strand 5 to 3 direction, just like you are remembering so many factual information. Just mug up in PCR if they want you to tell the sequence of primer, you will write both strand in 5 to 3 direction, and the initial nucleotide sequence would be nucleotide sequence of primer. So in this answer is, please do reply. So answer is D. You can also calculate, yes, if you want you can also calculate, yes, on this strand synthesis will occur in which direction, 5 to 3. The primer would be T2 A G2C A C A T T T. Yes, this is same, A C A C A T T T. So primer for this is, this strand is this. So again I say, don't get confused. If you want to calculate, you will try to calculate. I got very less answers. I know this is a difficult part, but remember the mnemonic. Now we are coming to next question. Biology